Respected Professor J.D. Khetkar, Coordinator, UGC-HRDC, Dr. Baba Sahib Ambedkar, Marathwada University, Aurangabad, Maharashtra, and the entire participants of refresher course in Biological Science and Technology. I am Mr. Satyam Tagam, roll number 22, going to present seminar on the topic Cytotoxic Study of Cestrum orienticum. I would like to start with the rationale of the study. Cestrum orienticum, a family Solanaceae, grows as garden plants and has become escaped an invasive species in many temperate regions of the world. This picture shows the twig of Cestrum orienticum with flowers. Its common name is orange jasmine. Cestrum orienticum possesses some medicinal properties. It possesses anti-inflammatory, antipyretic, insecticidal, anti-allergic, astringent, antibacterial, and antifungal, anti-HIV-1 and antioxidant properties. All these medicinal properties are induced by the different chemicals found in the plant. They are volatile oils, terpenes, steroidal alkaloids, flavonoids, saponins, phenols, and ligands. Due to the random consumption of this plant for medicinal purpose and its invasive nature, it has induces some kind of toxicity and fatalities in the different species, including plants as well as animals. Objectives of the study. There are three objectives of this study. Number one, to investigate the cytotoxic effects of aqueous extract of Cestrum orienticum on dividing cells of Ileum sativum root tips with numerical analysis of mitotic index and abnormality index. Number two, to know the different types of cytotoxic effects on dividing plant cells by the aqueous extract of Cestrum orienticum. Number three, to compare the patterns and status of cytotoxic effects of aqueous leaf extract of Cestrum orienticum on root tip cells of Ileum sativum as per the variation in the treatment concentration. Key definition used in this study: mitotic index. It is a percentage of dividing cells per observation. Abnormality index, percentage of abnormal cells per observation. Phase index, it is the percentage of individual phase of cell division per observation. Cytotoxic, it means toxic at cell and chromosome level. EC50, it is the concentration of any substance which bring 50% change. Materials and methods adopted in the present study. Materials. Source of aqueous extract is the Cestrum orienticum of the family Solanaceae, and the test plant species on which the aqueous extract has been treated to observe the variation, that is mitotic index and the abnormality index variation, is the Ileum sativum of the family Liliaceae, whose chromosome number 2n equals to 16. Methodology. One of the important methodology adopted in this study is the Ileum test proposed by Levan. In the first step of this study, rooting of Ileum sativum was conducted. 55 pieces of Ileum sativum cloves kept for 2 days in distilled water for proper rooting. And next is the preparation of 1 to 10% leaf extract Cestrum orienticum. In this process, 5 grams of fresh Cestrum leaves crossed in 30 ml distilled water and filtered with Whatman filter paper. Final volume of supernatant was made 50 ml, that is, it becomes 10% solution, and it is kept as a stock solution. From the stock solution, 1 to 9% solutions were prepared by the serial dilution method. Next is the root treatment. In this step, 1 clove in each solution, that is 1 to 10% solution, were kept with 5 replica for 18 hours. And besides, one control set, that is allium sativum rooted allium sativum, were kept in distilled water with 5 replicas for 18 hours. Next is the fixation. Root tips from all the treatment, including control sets, were taken and fixed in the mixture of acetic acid and ethanol in the ratio of 1:3 for 24 hours. Staining was done with 2% acetoarsine, and squash preparation was made for the each treatment, including control. And finally, the observation. Dividing root tip cells were observed and 10 microphotographs were taken for each treatment for the analysis of mitotic index and abnormality index. Under microscope, different types of chromosomal abnormalities were observed. So among them, important abnormalities were anaphase laggard. So here in this diagram, you can see two groups of chromosomes are moving towards two opposite poles, and few chromosomes are lying in the middle portion. So these are anaphase laggard. And another is the anaphase bridge. Two poles of chromosome are linked together by the bridge-like chromatin structure. Sticky metaphase. The metaphase chromosomes are intermingled to form the clumped structure. These are sticky metaphase, binucleate cells, micronuclei, and nuclear dissolution result. So in this table you can see, with the increase of concentration of aqueous extract of Cestrum orienticum, there is a regular decrease of mitotic index from 79.61 to 40.43. In case of control set, the mitotic index is 85.07, which is very high in comparison with the treatment cells. In the column of abnormality index, you can see here there is no abnormality index in control set, but with increase of concentration of aqueous extract of Cestrum orienticum, abnormality index was increased from 0.85 to 16.10. This graph shows the pattern of variation of mitotic index with reference to control 1, with the increase of concentration from 1 to 10%. And this graph shows the variation of abnormality index observed with the increase of concentration from 1 to 10% with reference to control set. This table shows the variation observed in the phase indices with the increase of concentration from 1 to 10% with reference to control set. Interphase index was found to be increased with the increase of concentration from 1 to 10%, that is, in 1% it is 20.39 and in 10% it is 59.57. Similarly, prophase index was found to be increased with the increase of concentration from 1 to 10%, and the metaphase index, anaphase index, telophase index shows the anomalous pattern of variation. In the control set, there is a regular changes or variation in the interphase index, phase index, metaphase index, anaphase index, and the telophase index. interface index is very high in control set, that is 14.8, whereas telophase index is only 2.05. And this graph shows the variation of the phase indices as per the change in the treatment concentration with reference to control 1. This graph shows the EC50. EC50 observed in this study, that is 7.6% of the aqueous extract of the cestrum orienticum is sufficient to bring the 50% changes in the, or the 50% chromosomal abnormalities in the ileum sativum root tip cells. Findings. From this study, I have got some important findings. So these are summarized in this slide. Number 1, with regular increase of concentration of cestrum aqueous extract from 1 to 10%, the mitotic index was found to be decreased. Number 2, with regular increase of concentration of cestrum aqueous extract from 1 to 10%, the abnormality index was found to be increased. All the phases of mitosis show chromosomal abnormalities. Cytoplasmic and nuclear degradation were also observed. According to phase indices variation, the maximum cells were in prophase and then interphase, metaphase, anaphase, and telophase in the control set, whereas treatment sets prophase and interphase were found to be decreased and increased respectively as the concentration increases. Other phases shows variable pattern of changes as per the concentration change. As per cytological abnormality, the most affected phase were prophase, metaphase, and anaphase. Conclusion. From the present study, we could know that a bioactive compound present in the cestrum, which are extracted in the form of aqueous extract, are cytotoxic, can cause chromosomal abnormalities, and act as mitotic depression, whose authenticity must be investigated through more refined molecular techniques and rectified protocols. With this, I would like to conclude my seminar presentation. Thank you very much.